Hello everybody and welcome to the latest in the SCI Fine Chemicals Group and Young Chemists panel webinar series. My name is Nessa Carson, so I'm on the YCP representing the Young Chemists panel. And with me I have to moderate the Q&A sessions Jason Tierney and Caroline Lowe, who are both from the Fine Chemicals Group. So these two panels both exist just to facilitate links between organic chemists in industry and academia, and also to provide a forum for discussion and sharing of information relevant to all aspects of pure and applied synthetic chemistry. So you've probably noticed by now that you are all muted. There will be a Q&A session at the end of each of the two presentations today. And to ask a question, please type into the questions box at any time during the presentations or during the Q&A session. I'd definitely like to thank our sponsors. So our series sponsors are AstraZeneca, GSK and UCB Pharma, and particular thanks to our one-time webinar sponsor, Liverpool Chirochem, who I believe are also sponsoring next week's webinar. So a little about that webinar. Next week, then, we're really excited to host a showcase of the National Retrosynthesis competition. So this is an annual competition. It's organised both by the SCI Fine Chemicals Group and Young Chemists panel, and also by the Royal Society of Chemistry, the Heterocyclic and Synthesis Group. So next week, we're going to show 3 of the winning teams, who are shown here from University of Leeds, from GSK and from Eutectic. And they'll be talking about their ideas to design a synthesis for this molecule neodyterpenoid A, which looks very, very challenging to me. So I'm really interested to see what they've done here. So please tune in for that next week. The week after, then we are featuring Neil Stropman and Anya Frischkowska, sorry, from Merck in the US, and they are presenting synergistic aerobic oxidation with a metalloenzyme and a transition metal activator or electrocatalyst in the synthesis of the key aldehyde intermediates in something. And development of an enzymatic cascade as the manufacturing route. So I think they're both presenting on the same synthesis, I believe, here. Well, without further ado, I'll introduce today's session. So we have 2 presentations today. I'll introduce each speaker in turn. And first of all, I'm very excited to introduce my former colleague, Scott Bagley from Pfizer. So he is going to be talking about palladium-catalyzed preparation of a cyclic alkenylsolfonyl fluorides, a new motif for sulfur fluoride exchange. Let me just talk through his bio a little bit. So Scott's a synthesis group leader in the inflammation and immunology group in medicine design at Pfizer in Groton. So he graduated from Bucknell University in Pennsylvania in 1989 with a BS degree in chemistry, and worked at Merck for a little while before moving to Pfizer, where he's been for 26 years, involved in many project areas, delivering over 10 clinical candidates. It's incredibly impressive across multiple disease areas. He's also authored over 35 publications and patent applications, and has established collaborations with academic groups at UC Berkeley, University of Oxford, University of Wisconsin, and the Hong Kong University of Science and Technology. In 2013, Scott received the Technical Achievement in Organic Chemistry Award from the ACS and continues to volunteer with the ACS, both as a member of the 2019 NOS committee and the DOC Virtual Symposium Committee. Anyway, his talk today is on palladium-catalyzed preparation of cyclic alkenylsolfonyl fluorides, a new motif for sulfur fluoride exchange, and I will hand over to you, Scott. Thank you.
Thanks, Nessa. I appreciate the invite from from the SCI and the Young Chemist group. Thanks to Theo and Oliver behind the scenes, and Jason, Caroline, and yourself for running today's session. Really appreciate the invite, and let's get started. We're going to talk about some alkenylsolfonyl fluorides. This is work that was done in collaboration with the Willis Group at Oxford, and with the grad student Terry Liu, who did a lot of the work. But I'm going to run through a little bit of sulfonate chemistry first, talk about why we're interested in sulfonates and sulfonyl fluorides, and how we got interested in some vinyl sulfonyl fluorides, why we like the motif, how we can access them through a broad ketone monomer set, and the multiple vector functionality that we found once we developed these fragments. And without further ado, I'll just introduce myself, I should say Groton a little bit. So a lot of you from the UK and other countries, not the US. So where I am right now is in Groton, Connecticut. It's about halfway between Boston and New York. We are right across from a city called New London. This is our site here, and my office and where I'm sitting right now is right about where this star is. And this is the Thames River, because we're in the US and somebody changed it years and years ago. And right up the river is a submarine base, and so about 2 or 3 times a week we get to see the subs coming in and out. This one's about 350 feet long, about the size of a soccer pitch. And right around the corner from Groton is a little town called Mystic, where they filmed Mystic Pizza, which is a famous movie with Julia Roberts and things. And there really actually is a Mystic Pizza that you can go to and enjoy. Anyways, just want to get started now about, as Nessa said, I've been in discovery and medchem for quite a few years. We're constantly looking for gaps in our synthetic methodology, and several years ago, one of these we identified was in sulfur-6 chemistry. While there's lots of traditional reactions in this space that have been known for a long time, their use has been limited to by functional group tolerance and other factors such as template availability, and basically the oxidative conditions you need to go from say a thiol you prepare from a SNAR or a coupling reaction is not necessarily compatible with late stage functionality of some of the molecules we're working on, nor is using metals, aryl metals or alkyl metals with SO2 gas, or this reaction here, which is refluxing chlorosulfonic acid, works fantastic, but it's obviously not amenable to many substrates, and also for library production. If we want to look at making hundreds of compounds at the same time, it's not something you really want to run a whole lot. It is done on an industrial scale, but the regioselectivity is controlled by electrophilic aromatic substitution rules. So that limits the amount of types of compounds that you can make. So we were seeking new reactivity to access the wider available monomer class, whether it's for parallel synthesis or scale up. And over the last few years, we've seen a lot of advances in transition metal catalyst and reagent design that I'll cover in just a moment. So one of the things I was involved in when we first got started with this was a collaboration with the TOST lab at the University of at Berkeley, where we invented a reaction to go from a wide monomer set of aroboronic acids with alkyl halides. That was a gold catalyzed with potassium metabisulfite as the SO2 source. And we were able to get catalytic turnovers, and with this new reaction, they weren't terribly high turnovers. We're talking about less than 10 turnovers. These are 10% catalyst loadings, and the reaction was not the best in scope, as we needed electron-rich aryl rings to drive the reaction. As you can see, a electron withdrawing motif gave us 0 yields. But we showed that we were able to get a broad reactivity with electron donating groups. Now, the reaction itself wasn't. We used it a few times in preparation of library chemistry, but the main learning from this reaction was actually the catalytic cycle that we developed and we proposed for this reaction. And what that does is it starts with the gold chloride pre-catalyst and a ligand exchange with methoxide to give the active, to enter the catalytic cycle, transmetallation with your aroboronic acid to give the gold arrow complex shown here. Insertion of SO2 into the gold arrow bond to give the complex shown here, and then regeneration of the gold methoxide from the solvent releases sulfonate as a reactive intermediate that can be then further derivatized to give sulfonyls, and other SO2 derivatives. Now, this is the most, to me, this is the most important part of the entire reaction right here. When you look at a traditional aminocarbonylation, for instance, you enter with an aryl halide, carbon monoxide, and an amine, and you end up every cycle you're spitting out a single compound, a single product. But over here, what you're doing is you're actually spitting out a key intermediate that you can then functionalize. And so that's where this sulfonate chemistry really shines. And so we can take that and do libraries such as this with this indazole. We can do alkyl libraries to give sulfones or go directly to sulfonamides. And so what you have then is a broad set of chemistry that's been really pioneered by the Willis group. We've done a bunch, the TOS group I just talked about, Professor Wu from Fudan University, and so many others, where you can actually get sulfonates from so many different sources using transition metal chemistry, some SN2 chemistry. The discovery of DABSO and utility in this reaction is has really blown this wide open, and the products you can make from these sulfonates are just impressive in their breadth. And now what we've been looking for is a way to access aerobromides in this reaction as a much broader monomer set. As you can see here, that sulfonates are not as well supplied commercially as, say, aerobromides. I also want to shout out to the folks at Merck and the Cornell lab for some beautiful work where they take sulfonamides and go back to sulfonates. Please have a look at that. That's really great work there. So with the Willis lab, we use this we developed this transition metal catalyzed, palladium catalyzed reactant to make sulfonyl fluorides using DABSO and the amphos ligand, which work beautifully to insert SO2 with isopropanol solvent and reductant to give and quench with NFSI to give sulfonyl fluorides. And so when you look at all the variety of sulfonyl fluorides we're able to get from aerobromides, then this you can see the utility in this work, where you're doing a late stage sulfonylation to access some of these reactive and important intermediates for chemical biology or other types of chemistry. Also like to shout out to the Ball Lab, an undergraduate group that I'll touch on a couple more times today, who published a very similar paper right after we did with just a super group of folks there in the Ball Lab at Pomona University, California. So why sulfonyl fluorides? So it's a strong sulfur fluoride bond. The SN2 reactivity is quite limited unless it's complexed with either in aqueous or in the inside of a protein, sometimes with Lewis acids I'll touch on in a moment. One of the largest uses of sulfonyl fluorides is for these sort of clickable sulfonyl fluoride probes, where you bind to a protein and then use an alkyne to tag on a marker, and you can pull those down for all kinds of chemical biology uses. Also, sulfonyl chlorides, well, they're super reactive, but they tend to react very quickly with nucleophiles. They're amazingly good for making sulfonamides and other important motifs, but they suffer from a reactivity where they can either be overreactive and they can hydrolyze very quickly and they're prone to decomposition on storage, or you can react at the chloride as well, so you can transfer the chloride instead of the SO2 group. Sulfonyl fluorides are much more stable, but recent advances from Pfizer and the Ball Lab I just mentioned have really opened up a lot of uses for sulfonyl fluorides in a library chemistry format, which would be when you reacting sulfonyl fluorides and amines in the presence of calcium trifluoride, it really pushes that reaction where you can see some of these amides, or these sulfonamides were made in high yield versus with and without calcium trifluoride. You can see the huge increase in reaction yields. And also recently they threw out a new paper where they're using Dabco as an additive, and that really knocks that down so these reactions run at room temp within hours. So we were looking at the next generation of sulfonyl fluorides, and these compounds, these ethylene sulfonyl fluorides, have been really around. They've been around for a while. They've caught a lot of attention in recent years. Barry Sharpless called these the most perfect Michael acceptors ever found. They react almost exclusively at the carbon of the vinyl group here, and then leave the sulfonyl fluoride for later functionalization. And the selectivity and reactivity of these make ethylene sulfonyl fluoride a really nice addition to the toolkit of both synthesis for chemical biology, but also just for regular compound synthesis. I stole this graphic from a nice paper, a nice review in Synthesis, where they talk about all the different types of chemistry that can be done on these ethylene sulfonyl fluorides, mainly at the carbon, at the vinyl portion of the sulfonyl fluoride, and you can see the wide variety of possibilities there. So we were wondering if we could take our sulfonyl fluoride methodology and expand that into some of these custom vinylic sulfonyl fluorides and see what do we get out of these? What are the products that we get? How can we use them? What's their reactivity? Is it going to be changed? Are we going to be able to divest that activity and focus more of it on the SN2 with the fluorides, or would we see reactivity at the olefin and such? So these had not really been explored in the literature at all. So we thought we were in a good position to have a look at these sulfonyl fluorides, these vinyl sulfonyl fluorides, and could we exploit those for a number of uses? So so this is where the Willis Lab and Terry Liu took this on, and I'm obviously going to summarize way more work than is shown on this slide here. This is, you know, a year and a half or more of a lot of effort from Terry, and I applaud his work here, where we took our initial conditions of palladium acetate and the potassium catalyst, where on cycloheptene triflate, I'm sorry, iodide, and they were only seeing about a little bit of conversion to the sulfonate and then the sulfonyl fluoride. One of the things that we're seeing, and this kind of plagued it a bit, was that these sulfonate intermediates were quite reactive, so actually lowering the reaction time helped increase the overall yield. Terry noticed quickly that the isopropanol in the first step of the reaction inhibited or actually reacted further with the sulfonyl fluoride that was prepared, so the yields dropped. So switching solvent away from isopropanol to other solvents in the second step boosted the yield quite dramatically. And then revisiting our favorite amphos catalyst allowed us to access bromides, and then switching to triflates really is where it started to take off. We started to see that here we're getting into quite usable yields with the triflates, and that's where we started to say, well, these are, you know, the monomer set of enolizable ketones that we can convert to triflates is just much broader, so we can really access a lot of compounds in this area. And so taking a piperidine ketone here, piperidone, and converting to triflate in high yield, and then a 90% yield to the vinyl sulfonyl fluoride using the conditions outlined here with ethyl acetate and the second step, was a real good indicator that we'd have a lot of success in this area. So you can see a whole bunch of compounds that were prepared with this new motif, and with a wide array of substitution on the cyclic motif What you will not see, and this is where I think the Willis lab is really pushing forward in here, is the reactivity of both the triflate and the sulfonate intermediate limited some of the success, and so they're searching for some alternate conditions to get some, to access a lot of these alternate motifs that we were not able to successfully produce in the first go around. But some of these are quite reactive. So like for instance, this one here just is going to be something that tough to isolate. But one of the most interesting, as a side note, one of the most interesting findings was that the aryl triflates did not react one bit. So if you just take this biphenyl triflate, there was no reaction under these conditions. And if you take a triflate here of this half saturated system, you can get a good yield of the sulfonyl fluoride. But if you have a triflate on the aryl ring, that one is completely untouched. And so there's some exploration that the Willis lab's looking at this point. That going forward, I'm sure you'll hear about this later on. That's some interesting finding. But what I'd like to get into is the reactivity of some of these vinyl sulfonyl fluorides and the utility that we're seeing for both synthesis and chemical biology. So taking the piperidine, we can actually functionalize in a number of ways off of this piperidine, where you can deblock with HCl or TFA to get a bench stable solid that's you're able to functionalize through amide couplings, isocyanate reactivity. You can prepare all kinds of. This one is a UV vis label that you can use for chemical biology tools, or a biotin tag. One of the interesting reactions here is you can do a reductive amination right here on the piperidine without affecting any of the other functionality in the piperidine. In addition, the N-benzyl can be you can exchange the protecting group to get this alkyne installed, and then a click reaction shows the utility and of this kind of tagging motif in these compounds. Now you can actually start with do the whole reaction from the triflate with the alkyne in place, but it needs to be protected, and it's a decent usable yield, but not as high, but shows that you can actually start with the alkyne in hand and have a really nice it's almost triple headed motif for compound for whatever you would like to use it for. Now, as far as the olefin goes, I'm sorry, the sulfonyl fluoride goes, you can react at the sulfur in an SN2 format, which was quite interesting because you don't see this in the vinyl sulfonyl fluoride motif, the original just ESF molecule. You see only reaction at the carbon, but in these cyclic group, you actually see a differentiation in reactivity at the carbon versus the sulfur. So you can get O displacement, you can get sulfonamides directly in moderate yield, but you can see that's a very high loading of a very nucleophilic amine. If you use the calcium trifluoride conditions from the Ball lab in Pfizer, you can actually increase the yields on quite non-nucleophilic anilines, as shown here with paratoluidine and aniline, giving reasonable yields in much about the same time frame. So what you're seeing here is that these motifs in a chemical biology sense could be used to mark proteins and peptides that have nucleophilic residues such as tyrosine and lysine. So you're talking about getting some nucleophilic substitution at the sulfur. And but when it comes to the olefin, there's a lot of it. I showed a lot of reactions earlier that one could possibly do. So we've explored some of that work here. Hydrogenation only touches the olefin here, and it allows you to access fully saturated sulfonyl fluorides. The utility of those is becoming more well known, but a nice access to a very unique motif there. And then reactivity at the olefin, we did not see very good reactivity with amines at the olefin, but with sulfur, there was almost complete Michael addition into the olefin to give the sulfur connected analogs, whether it's a aryl or captans here. The cis-trans ratio wasn't all that great, and there was some, not sure why there was a mixture of cis and trans. I'm sorry, I had these as DRs, but it shows that we can get cysteine nucleophiles from proteins to react at this carbon. So cysteine actually works really well here, but the product was a wild mess of diastereomers, and so but so it wasn't reported in the paper, but the reaction actually went in quite high yielding, almost quantitative. You can see where this kind of motif mimics one of these covalent type kinase inhibitors. So you can imagine using some of these molecules in that type of motif, in that type of drug-like molecule, or in sort of a chemical biology fashion to try and mark these proteins. So we have so looking at these sulfonyl fluorides, these vinyl sulfonyl fluorides, these cyclic alkenylsolfonyl fluorides, we've shown that there's reactivity at the sulfur. We've got reactivity at the carbon with the sulfur nucleophiles. We've got the ability to deprotect and modify the other end of these molecules. And we can access a lot of these from readily available ketone monomers. And there's a lot of more work to do in this area, so we can look at both the alkyl and other ketone starting materials that didn't perform in this first round. But I believe that this is a nice addition to the toolbox for both synthetic chemistry and to chemical biology. We're able to access a lot of utility and variety in the reactivity of these new handles. And I would like a huge shout out to Professor Willis for the work there, and his involvement in this collaboration. Terry Liu for his persistence in pushing this chemistry forward. I'd like to also thank Dr. Alan Davies, who was in the original sulfonyl fluoride paper that we've actually hired here at Pfizer a couple of years ago. So thanks to those guys. I would like to shout out to Professor Ball and his lab at Pomona. Wonderful work coming out of that primarily undergraduate institution. They've done a lot of great work with Pfizer on work with sulfonyl fluorides. I'd like to thank Professor Tost and Miles Johnson, who was my co-worker while he was a grad student in the Tost lab on the original gold synthesis. And a huge thanks to all the folks, and this is a very abbreviated list of folks that have been involved in the SO2 work here at Pfizer. So looking forward to a lot more in this area, and really happy to have been involved in this work. And thank you to all of you for listening. Thanks to the organizers for the invite, and happy to take any of your questions. And I think we're right about on time. Thank you very much.
Fantastic talk. All right, I think we should have 5 or so minutes of questions now, and then if you're happy to stick around, then we may be able to get in a few more after Richard's talk.
Okay, so I'll start off. So we got a question from Paul Colburn from Liverpool Chylochem LCC. So he's got a question about whether the sulfonyl fluorides have been successfully applied into any DEL synthesis, so doing library, DNA encoded library synthesis, which you mentioned about the sort of proteomics.
Right, so I have not done any the sulfonyl fluorides and in the DEL area. I'm personally not involved. I don't know of any specifically at this point. I do think that the addition of the ability to use that calcium trifluoride to really soup up the reactivity of the sulfonyl fluorides, in addition to the fact that you can make and store these sulfonyl fluorides without a whole lot of worry about them degrading over time, it's going to really make them a much more useful alternative to sulfonyl chlorides. But yeah, we've started to look into a much broader use of sulfonyl fluorides here at Pfizer in our library synthesis. You know, some of this, especially the calcium trifluoride stuff, is brand new to all of us. So we're really looking hard into it as to how we can best use it. But it's going to be something that's really useful. That's for sure.
Yeah, I'm sure Paul would like to speak to you about that in the future, Scott.
Absolutely. Please reach out anytime.
Caroline, do you want to continue?
Yeah, sure. So we have a question from Ian Clark, who says, on page 15, slide 15, why was the reaction less successful when you ran it with other ring sizes or open chain systems?
So it's a good question. Thank you. And one of the observations that Terry had when he was running a lot of these was that there was degradation of the reaction components, whether it was at the triflate stage, maybe it was a more unstable triflate, or the sulfonate. A lot of times the sulfonate was formed, but then would decompose rather quickly. I think it was just up to there was some. We haven't figured out any of the actual full-on reasons yet, but there was definitely a set that were overreactive or at the triflate stage, some at the sulfonyl fluoride stage, and some at the sulfonate stage. And there wasn't a whole lot of classifications that we could make and say that this set fell apart at the triflate stage. That's one of the things we're really looking, I should say, Mike's lab is working on really hard on right now, is to try and broaden the scope beyond these ring sizes that we see here, because we would really like to get into a lot more.
So I noticed that you've gone back to isopropanol there. Is it a solvent effect?
So the isopropanol is used for the sulfonate installation, so triflate to sulfonate, and then switch to a non-nucleophilic solvent for the fluorination stage. So that was another big one, is that some of these sulfonyl fluorides were super reactive. So any trace of isopropanol left, or maybe a little water or something, they just hydrolyze, or they react and go off. So some of them are just overly reactive.
Okay, that's quite interesting, because we've got a similar theme. So there's a question from Aaron Atka, who's from GSK in the UK, and he's got, have you explored the hydrolytic stability of these cyclic alkenylsolfonyl fluorides? If so, at what pHs, and to what extent are reactions with various nucleophiles chemoselective in aqueous solvents? He gives an example there of a conjugate addition versus substitution at sulfur, and he comments on mixtures.
That's a great question, and yes, when you start getting into chemical biology, now chemical biology is not my field. It's just I really like these motifs, but we have not yet studied that, but we have discussed with our chemical biology team here at Pfizer, and there are some studies going on at this point, but at this time I don't have any I can't say that I have anything that I can tell you about that at this point, and we didn't study it in this publication.
Okay, thanks for
sorry for that. That seems a cheesy answer, but no, we didn't study it, and we are looking into it in-house though.
No, it highlights it's a very good question, Aaron.
Yes.
Just can't comment on it. I've just got one follow-up question. So it's with the so there's been some work done in the past by Steve Caddick using pentafluorophenol esters as an activating sulfur group. So I just wondered how they would compare with this sort of chemistry, especially where you're doing your quite interesting sort of palladium coupled chemistry.
So as a triflate alternative, I know that we did look at a number of different sulfates, phosphates, etc. Triflate was the one that really worked. I know Mike has done some work previously with the pentafluoroesters. I don't think that worked in this case. Now,
okay.
Also, some of Mike's work, some other SO2 papers was using the pentafluorosulfate ester in place of, say, the fluoride, and that's a quite useful leaving group for other functionalities. So that could be something that might be useful in a later stage diversification instead of the fluoride. But as far as the like the alkenyl replacement for the triflate, yeah, the triflate was really the only one that we got to work.
Yeah. Okay. Have we got time for one last question?
Maybe one more.
Okay. Comment here from Chris Brown, who says, "Hi, nice talk. What does the calcium trifluoride do?" Chris Brown from Eutectic.
Beautiful question. Yes, thank you. So calcium trifluoride is what it does best is it actually gives a good tug on the fluoride. So calcium fluoride is extremely insoluble, and so in addition to being the calcium trifluoride being a nice Lewis acid coordinating the fluoride, the precipitation of the calcium fluoride really helps drive that reaction. So you're giving it a good tug, you're getting it out, very much like silver with your halogens, and allows this to go move quite a bit faster, and it pushes the equilibrium all the way, pushing it all the way to the sulfonamide. The Dabco is a really nice addition there, and some of these reactions go room temp in less than an hour, and it's very much like adding DMAP to an amide coupling. It really gives it just a bit of a boost, and that was a really great find.
Great. Well, thank you very much for that talk. I'll introduce our second speaker of the day now. So very excited to have Richard Horan from GSK. Richard obtained his master's degree in chemistry from University of Southampton before moving to University of Cambridge for his PhD with Darren Dixon on the asymmetric synthesis of amino acid derivatives, which brings terror into my heart because I've had to work with amino acids and their asymmetric synthesis in the past, and it's terrifying. So he went on to do a postdoc with Steve Lay on synthesis of natural products, including the benzyls and spongistatin, before joining GSK as a process chemist in 2007. Since then, Richard's worked on a wide variety of both early and late phase development projects, the development of enzymatic transformations as part of the SynBio group, and roles in outsourced project management and outsourcing strategy. Richard is a GSK Associate Fellow in the Chemical Development Department and is currently working as a chemistry lead on a late phase project. So today he will be talking about control in process development, evolution of the manufacturing route to vestipitant. Thank you very much, Richard. I'll pass over to you.
Excellent. So thank you for the kind introduction. Thank you to the SCI Young Chemist Panel and Fine Chemicals Group for the invitation to speak today. As mentioned, I'm going to speak a little bit about a project I've worked on a couple of years ago, vestipitant, and specifically looking at control in process development. And so I'll come to what that means and what we looked at. There's lots of bits of this project that I won't be able to cover today. If there's anything in particular that you're interested in, ask me and during the questions, and we'll see what we can do. So a brief introduction to vestipitant as a molecule structure on the top right of the screen. It's this urea functionality in the middle that couples two chiral benzylamine parts, and the API that we were making was the salt with methane sulfonic acid. It's an NK1 antagonist, and we were interested in at the time as an antiemetic, although it's also been investigated for a couple of other indications. Again, we were interested in it for IV delivery in a hospital setting, and the proposed manufacturing site was Singapore, and the significance of that will manifest itself a little bit later. So the synthesis of vestipitant is, I don't think, going to surprise anyone. So you can disconnect around the central urea fragment to give three fragments, the diamine on the left-hand side, some kind of one carbon linker unit in the middle, and then the trifluoromethyl benzylamine derivative on the right. The benzylamine derivative could be further disconnected back to the ketone. That's widely available on the market. And the diamine fragment on the left can be further disconnected back to an ethylene diamine, some kind of dicarbonyl unit, and the aryl bromide as a grignard precursor. So I'm going to go through the route of manufacture as we inherited it. There'd already been some work done by my colleagues in Italy, our site in Italy, and there's some publications links down the bottom if you're interested in theirs. They did a lot of fantastic chemistry, and actually there's a number of pieces of chemistry that I'm going to put up on the slide in a moment where I look at them and I think, wow, I wish I'd thought of that. I'm going to go through this pretty quickly. I will throw up this scheme a few more times during the presentation and talk to specific parts of it. So don't try and take it all in, just enjoy the ride. So their synthesis started with this oxalamide derivative and the corresponding Grignard reagent to give the aryl ketone here, which could be reacted with ethylene diamine and then hydrogenated to give the racemic amidoamine, as shown here. This was then one of the first pieces of chemistry that's really beautiful. There was then a dynamic kinetic resolution of this, so the aldehyde is just there to acidify the proton at the chiral center, set up a fast pre-equilibrium from which the desired isomer, stereoisomer precipitates out as the mandelic acid salt. From this stage, we then reduce the amide using borane generated in situ, and then selectively Bock protect the least hindered amine to give us one half of the molecule ready for union with the other half. So starting from the benzylamine, we can treat that to generate the carbonyl chloride using, again, some interesting conditions. So we actually use carbon dioxide as the one carbon unit here, together with a few other reagents to convert that through to the carbonyl chloride, which is actually remarkably stable in solution. That can then be reacted with the other amine fragment to generate the protected API, which is then deprotected and crystallized to give the API as its methane sulfonic acid salt. Very briefly speaking to the benzylamine derivative, as I mentioned, that comes from the aryl ketone, and just through a reductive amination, and then on this time a classical resolution to give the salt, and that can then be free-based, ready to go into the next stage. So when we inherited this project, we were looking to go into phase 3 studies, at which point we start thinking about how we're going to put together a control strategy, which is the set of controls we're going to use, and we are going to demonstrate and prove work that we are then going to commit to the regulator that we will put into place during routine manufacture once we launch our product onto the market. And so in developing that control strategy, what we do is we start with all of the knowledge we have about the chemistry, our experience from the lab and from plant campaigns, any theoretical understanding we might have, any published information. And from there, we start to identify what are the critical quality attributes, what are the attributes of the API that impact on the patient. And usually, and I'll speak to this in a couple of slides' time, but usually that's things like purity, specific impurities, physical form, and so forth. Where do we control those? Where are any impurities formed or introduced? Where are they controlled? And what levels do we see against what levels can we tolerate? And it's very much a risk-based process. The higher the levels, the lower the tolerability, the more likely we are to define something as critical. If we have really good control already, the levels are very low, or we can tolerate a much higher level, then that's probably much lower risk and therefore maybe not a critical quality attribute. From there, we can start to develop or establish or demonstrate existing or new controls, and those can be procedural. So, for example, if running the chemistry in, let's say for a random example, DCM gives you an impurity, but running in toluene, you never see that impurity, switching from DCM to toluene would be a procedural control. It's a one-off sort of binary on-off switch that allows you to turn off that impurity, whatever it is, and that's usually the best control you can get, and is the one we will aim for wherever possible. Failing that, we can either rely on attribute control, which is things like specifications of intermediates, or parametric controls. This is process parameters, so temperatures, stoichiometry, things like that, keeping those within a certain range. And normally it'll be a mixture of those attribute and parametric controls that we will look to put into place if we can't put procedural controls into place. I've put operational controls in brackets there. That's because these aren't controls that we would normally declare to a regulator. And operational controls might be things like if you are looking to control the level of inorganics in your API, then getting a good phase separation might be an operational control. It's not something you can really declare to the regulators that we're going to control this by getting a good phase separation. It's really dependent on the people and the process that they are operating on the day. So not normally where we would like to be, very occasionally we end up using them. Now, as you develop those controls, as you develop your understanding, you will get inevitably more knowledge, and that will feed back into your CQAs. It may be that actually you can put some controls into place that mean that your critical quality attributes become non-critical because you are routinely seeing them at much, much lower levels. They are no longer critical, and therefore you can describe less detail about them in your regulatory file. And at the end of all of it, so that can go around in a cycle quite a number of times, and in the end, what you write into your regulatory file is your control strategy, what you're going to control, how you're going to control it, and why you're going to control it, and why those work. Your design spaces, in the case of parametric and attribute control, what is the window you have to operate your process. And it's worth saying that not all of the knowledge that you have will go into the file. There's an awful lot of information where you'll often make a relatively high-level statement in the file, but then there'll be a huge amount of supporting information. And during the review of that file, it's very common for regulators to reach out to companies with questions and ask for additional information. And so having that at your fingertips is really important. So I keep talking about control, and I talked about critical quality attributes, and what I mean by this is things like the identity. I mean, it's a fairly obvious one. You've got to make sure that you're giving your patient the right material, but it's not about what you know, it's about what you can prove. And so, you know, proof of identity is an absolute must at every batch of material. The key one that we always look at is impurities. You know, as chemists, we're used to thinking about this, and we most often think about related impurities. So impurities that are formed during the chemistry that are related to the API structure itself. But we also need to consider genotoxins, metals and inorganics, solvents, including water, and bioburden and endotoxins. We also need to think about things like the physical forms, so the version, the salts and a sulfate polymorph, and habit, crystal size and shape. And it goes without saying, but it bears saying, that any material that is going into humans must be manufactured under good manufacturing process practice. Sorry, and the C stands for current. Today, limited time, I'm actually only really going to talk about the related impurities. I've got some slides on, I think, just about everything else. So if there's anything particularly you're interested in, do ask. As it happens for physical form, because it's an IV formulation, the drug product is actually vials of solution. So the physical form was very, very much less important, very much less impactful on the quality of that. And therefore, we focused on that less than we would of some projects. So thinking about impurities, these are just standard limits that come from ICHQ3, which is sort of an interpretation of the regulations that we in industry use to guide our development work. But for a drug where the maximum daily dose is less than or equal to 2 grams a day, which vestipitant was, the reporting threshold, so the point at which you have to declare a peak on the certificate of analysis, is 0.05%. The ID threshold, any impurity that you see greater than 0.10%, you need to know what it is. You need a structure of that impurity. And qualification, anything over 0.15%, you have to have qualified in a toxicology study. So toxicology studies are generally in vivo studies, they're animal studies, and they provide cover for impurities at levels greater than 0.15%. And we normally run these on API that has, representative levels of impurities in it. On this particular project, the tox studies had actually been run on material from a previous route, so different intermediates. And so as a consequence, it had a different impurity profile. And this is pretty common that we encounter this problem. And there is always the question that, some of the impurities are not covered by that toxicology study. And so we have to ask ourselves, can we control any impurities where we don't have tox cover to this 0.15% limit, or do we need to rerun the tox study? And the preference will always be to control them, because tox studies, because they involve animals, there's a really strong ethical drive not to have to run those again. So very briefly, in terms of how to identify the impurities and what impurities we should be looking for, clearly you'd look in your API, what impurities are there. You'd look in the mother liquor. So when you do your final crystallization, the supernatant that comes off that, if there are enriched particular impurities in that, those are being removed in that final crystallization. And so if something were to go wrong in that crystallization, they're the ones that are most likely to be present in the API. You'll also look at previous stages, both isolated and mother liquors. You'll use chemical logic. So, you can predict quite often what many of the impurities are, and you'll see on the next slide that those are probably quite clear to predict. And eventually what you'd be looking to do is do some spiking and purging work to look at what levels can be tolerated at each stage, where they're removed, how they react, and so forth. So for vestipitant, that's the structure shown in the box. We had 3 impurities that we believed were critical. The first 2 are diastereomers of API, so this is where our chiral center is of limited purity. And the third one, the diamine, is actually an intermediate in our synthesis. So speaking to those diastereomeric impurities first, as I said, they're just diastereomers of API, but they're enantiomers of each other. And when we inherited the process, there was very little purging within the stages. So if we want to control those impurities, we really need to control them in the starting material. So the stage 4 is the benzylamine, and then the stage 1, which I'll speak to a bit more in the next couple of slides. That was going to be difficult. I'll come back to that in a moment. The other thing that's worth noting here is that previously these were analysed as a single compound. So while we had tox cover for a peak, because they were coming from different routes, we didn't feel that that was actually scientifically we felt there was a hole in our scientific understanding there. We couldn't go back and reanalyze the tox batches to try and separate out these peaks. And so what we're really looking to do is to use those generic limits and avoid any need to rerun the tox study. So our overall strategy here was that we would build our understanding of the formation and purging of those impurities in stage 1 and stage 4. And we would build an argument to say we understand the chemistry so well that we don't actually need to test it in enantiomer. We know that it will pass spec in enantiomer because we understand the chemistry well enough. So looking at the stage 1 reaction, this is the reduction of the amide to give the amine. So you take a slurry of sodium borohydride in THF, and to that you add a solution of the salt in THF with a small amount of tetrabutyl ammonium bromide, and that's just there to solubilize the salt. You add that in. It's important this in solution from a safety perspective because you're adding a solution to a slurry, to a suspension, and it is reactive. It does give off hydrogen as the acid reacts with the sodium borohydride, but that's about the limit of all the reaction you see there. We then add BF3-THF complex, and that converts the sodium borohydride into borane in situ. That then does the desired reduction reaction, and at the end of the reaction we quench it with an alcohol, we distill off the THF, and then we add acid to crystallize out the bis-HCl salt of the product, and we filter that off and isolate it that way. A couple of things to note here. If you've not come across BF3-THF before, it feels about as reactive as BF3 etherate. It's just it doesn't fume in air. So rather than having to handle it with a syringe, you can just pour it out in a measuring cylinder in the fume hood. So it's much easier to work with. So I do recommend that. But it's not innocent. So if you do the reaction just with borane THF complex, which you can buy, it's very much slower. And so there's something going on during the BF3 addition that is making that reduction a lot faster. So in process chemistry, we tend to use something called design of experiments to optimize our reactions. And I just wanted to put in one slide to explain that for those who have not come across it before. So most people, university, are taught optimization to do one factor at a time, OFAT optimization. So let's say you have 3 factors you're looking to optimize: the amount of a reagent, the temperature, and the concentration of your reaction mixture. You might start off by studying different temperatures, find the temperature that works best. Then you would study different concentrations, find the one that works best, and then different amounts of reagent and find the one that works best. And that does work. But it doesn't tell you a lot about the rest of the design space, and it doesn't necessarily tell you where the optimum point is within that design space. And it's also not very efficient. You tend to require a lot more reactions to do this. The alternative is to do a design of experiment approaches where you take your design space, you do reactions at the corner of the corner points of those design space and at the center point of that design space. You might not do all of those. You might choose to remove some of them, but because of the statistics that underpin this, you can get a lot more information, potentially with a lot fewer reactions. And it gives you that systematic, thorough coverage of the experimental design space, which is great, and gives you really good understanding. And it's sort of there's an expectation in regulatory files these days that you will have these design of experiments approaches. So going back to stage 1, we ran a factorial design. So in the last slide, I showed you a 3-factor design. Here we're using a 6-factor design. So the amount of TBAB, the amount of THF, the equivalence of the reagents. So we've fixed the ratio of sodium borohydride to BF3, but we're looking at the total equivalence. Temperatures during the addition and after the addition, and the stir rate. Because this reaction is never completely in solution, we felt the stir rate was important to highlight any issues that might come back and bite us later. So we managed to study all 6 of these in just 20 experiments, and this was probably a week to 2 weeks work. So it's really fast and efficient reaction. And the half normal plot you see in the top right is a sort of representation of a statistical analysis, and it tells you which of these factors is having an impact on whatever you're measuring. So in this case, I'm looking at conversion. And so all of these factors shown kind of along this line on the left-hand side, when you vary those, it's not having any impact on the conversion. And it's only these that are further away, and the further they are, the bigger the impact. So C here is having the biggest impact. That's the number of equivalents of reagent. Now, that shouldn't come as a surprise. Let's be fair about this. If you're looking at the conversion, the more reagent you have in there, the more conversion you'd expect to have. So it's not a big surprise. Similarly, the temperature after the BF3 addition is significant. It's also worth noting here, this here is an interaction effect, and this is another expectation of regulators, is you understand how things interact. So I'll try to get this the right way around. At low temperature, the number of equivalents of reagent has a much bigger effect than at high temperature, and so you can see those interactions, and you can account for those in your design space. What this also showed was that curvature was significant. So this is not a linear response, it is curved. And although this particular design was not powered to model the curvature very accurately, for the sake of producing a pretty picture and my understanding and things like that, I did fit a curved model to it. This is not particularly accurate, but it's a useful visual representation of the data. And it shows, as you'd expect, higher temperature, higher equivalence of reagent, that drives up your conversion, and it kind of plateaus towards the top. Now, I've been talking about conversion during this slide, and actually what I've said is in the control, what we're really looking for is the control of the enantiomer. And as it turns out, the reason I'm talking about conversion is that we didn't find any factors that were significant in terms of controlling the enantiomer. So it's noise. There's something that we are not controlling there that is determining the enantiomer content of the product. We did quite a lot more experimental studies, and we never actually managed to get any control really during the reaction. But what we did note was something when we crystallized the product. We started to see that when we crystallized it from different solvents, we could see solvates. And this is where, when you crystallize the compound, solvent is incorporated into the crystalline lattice. It's filling gaps in the crystalline lattice, and methanol and ethanol formed solvates, and these actually gave really good purging of the enantiomer in the isolation. But if you formed an ansolvate from IPA or a hydrate, then they didn't purge the enantiomer. So we could aim for the methanol, and we started off this work with methanol, but we found actually the methanol solvate was unstable. It tended to lose methanol on storage. It became quite sticky, but it also converted to a sticky hydrate in a humid atmosphere, such as you might find in our manufacturing plant in Singapore. So this was actually, quite a big problem. But the ethanol solvate was much, much more stable, and that worked really well, and we were able to scale that up, good yields, and we got really good control over enantiomer. Now, I should have said on the previous slide, it's not just about, so we can't control this in the, I will go back, sorry. You can't control this in the input, because if you attempt to recrystallize the starting material, the input material that you're using, because it's not completely, that stereochemical center is not completely stable, whenever you recrystallize it, you always see some erosion of the stereochemical integrity. So it's not possible to get purity there. You also see a small amount of epimerization during the reaction itself. So we can't control it at the starting material itself. We can get limited control of the starting material. But what we're now seeing is control of the enantiomer level during the isolation. Although at 0.3%, we're targeting 0.15%. We need more control over this. So we next looked at stage 2. Now, in the original scheme that I put up, stage 2, this protected diamine product wasn't actually isolated. It was you got a solution of this and you took that straight into the next reaction. So we considered whether or not we could isolate this and therefore get another control point, another procedural control of this particular CQA. And we found that if you crystallized the free base of this product, you didn't see any improvement in the purity. However, if you crystallized it as the HCl salt, you did see actually really good purging of the enantiomeric impurity. And so we could take that through, scale that up, and get really good control over our enantiomer, well below our target levels. It might feel a little odd to make a HCl salt of a Bock-protected amine, but it was stable so the solid was nice and stable, and during the crystallization, it was stable enough for a period of several hours so that we could run the chemistry. So that worked really well, and we've now got really good control over that CQA. So moving to stage 4, the other chiral fragment in our molecule, the benzylamine here. This is a classical resolution through crystallization, and so as a consequence, we can just recrystallize this to get the purity we want, which is a very simple way of doing it. But in terms of understanding and being able to prove our understanding, what we used was a ternary phase diagram, as shown on the scheme here. So I'll speak through this very briefly for those who have not seen them before. These are basically, this triangle is a way of representing three-phase mixture or three-component mixtures. So at each point you have 100% of that component, and then anywhere else within the overall triangle represents a mixture of those three. So I've divided it into 4 parts. So this triangle along the bottom here, described by the blue line, up to C and down again. So within that, you've got ethyl acetate, undesired isomer, and desired isomer, some of it in solution, some of it out of solution. This top part from A quadrilateral here, just ignore the red line for the moment, is where everything is in solution. So as you've got more and more ethyl acetate, basically everything goes into solution. This triangle along this side here is anywhere in this point, all of the desired isomer is in solution, but some of the undesired isomer is out of solution. And what is most interesting to us is this triangle here, this black line, this blue line, and anywhere within this triangle, all of the undesired isomer is in solution, so it could be washed away, whereas at least some of the desired isomer is out of solution. So this is where we want to be operating our process. And this red line here represents a one-to-one mixture of desired and undesired isomers. So a racemic mixture. When you go in with a racemic mixture. And so you start off with that, and then as you add ethyl acetate, you will move up this red line. And what we want to do is we want to get to the point D, and this is the point where all of the undesired isomer is in solution, at least some of the desired isomer is out of solution, in the minimum amount of solvent, and this gives us the best recovery of our desired isomer. And actually, so in order to do that, what we need to do is to determine the eutectic point, which is labeled C here. And from there, literally, we can just draw a straight line to this point here. And you can determine the eutectic point with a single experiment. You make up a saturated slurry of material in a particular solvent at a particular temperature, let that come to equilibrium, take a sample of the solution, and that solution composition will be this eutectic point. So one simple reaction gives you that eutectic point, draw the line, find this point D, and from there you can calculate how much solvent you need, what your recovery will be. And that's brilliant. And so we did a few of these with different solvents, different temperatures, but very, very quickly you can determine your best point. We did do some more work to prove that was accurate. There are some assumptions built into this, but it's a fantastically powerful way of doing it. And I highly recommend this paper written by Clément with a number of the other members of the team, which describes this work. This is not my area of expertise, so if you have questions, I'll do my best, but I can't promise to know all of the answers. So one experiment to determine the eutectic point, and then a calculation. So now looking at the diamine impurity over here. So this, as I mentioned, is also an intermediate in the synthesis. And so this material in stage 2 is taken through to the selectively protected, coupled, and then deprotected. And we end up with some of the diamine left over in the API as one of our critical impurities. So during stage 2, inherently with any selective protection, you will get some bisprotected material and some unprotected material. And actually the unprotected material is not a problem. Both amines react in the next stage of chemistry. That impurity is really well purged, and it's not a problem. The bis-protected material is a problem. This does not react in stage 3b during that coupling, but is then deprotected in the stage 3c, the deprotection, and does give the diamine. However, now that we've introduced an isolation at this point, because this bis-block material can't form the HCl salt, it's very well purged in the isolation, and so we never see this material in isolated stage 2. And so it's very well purged. So that's not a source of this. The final place this can come from is obviously in the stage 3b reaction. If you have an incomplete reaction, you'll have some of this present, which will then track through into the product. Now you can try and drive this reaction to completion. You can use larger amounts of the carbonyl chloride coupling partner, and so that's a parametric control you can put into place. You can use a test to make sure you've got to low enough levels. That's an attribute control. What we chose to do is both of those things, but also we introduce a dilute acid wash at the end of the reaction, which protonates this material, takes it to the aqueous phase, doesn't impact on the Bock groups. This is unaffected, stays in the organic phase, and you can wash out the unreacted starting material. So we now have a procedural control, best class control we can have, and that gives us really good control over this impurity. I'm aware I'm running out of time, so very briefly about the aryl regional isomers. This is always something that regulators tend to be interested in because, and Scott mentioned it during his lecture, you're always reliant on the selectivity from aromatic substitution reactions, and so these are never completely devoid of the other isomers. So our desired bromide here is actually made from fluorotoluene through bromination, and so these are the impurities both from a theoretical perspective, but also if you analyze samples, these are the main impurities that you see. And we tested these in stage 0 chemistry. What it turns out, when you make the Grignard of these two materials, you actually see elimination to give the benzene. That reacts with the Grignard probably of this molecule here to give biaryl products. Those are readily purged. So actually we could demonstrate we had really good control over these potential impurities as well. And therefore we didn't believe they were CQAs. We did need to get some knowledge so that if we were asked by the regulators, we could demonstrate what was going on. So looking at the changes that we've made to our process, just in terms of quality. So in stage 1, all we've done is switch from a methanol to, this is industrial methylated spirit, so mostly ethanol as our isolation solvent to get that ethanol sulfate here. We've also introduced an isolation at this stage of the HCl salt to purge the impurity. As I mentioned, so we've got better understanding of the amine fragment, I suppose, of the salt here. So we've always got good control there. And we've introduced this dilute sulfuric acid wash to purge the diamine impurity at the end of that stage. So those are, some really small changes, but really profound impact on the quality of the material. There was also a whole bunch of other things we did. So we've simplified stage 4A. There's quite a lot of distillations in here, and so we're actually looking to reuse the solvent that we distilled out. That helps with our environmental stability and lowers our costs, which is great. Swapping triethylamine to tributylamine had a really profound impact on the rate of the reaction. I think from something like 16 hours to 1 hour. And we still don't understand why this was. So if anybody has any suggestions, I'm all ears. But a really great study, and we looked into the kinetics there. Some kinetic studies of the deprotection, that's largely around the safety from the gas evolution here. And then forming the salt here, we were looking at pH to control that charge, which is an interesting way of achieving that control there. So all it remains for me to do is to thank all the people who did this work. As with any work in industry, there's always an enormous team behind it. It's one of the things I love about working in industry is just all the very many really brilliant people you get to work with. So I'm extremely grateful to them for all of the work that was done here. Thank you again to the SCI, the Young Chemist panel, and Fine Chemicals Group for the opportunity to speak today, and I'll very happily take any questions. Thank you.
Thank you very much. Once again, wonderful presentation. I think we're very lucky to have had 2 great speakers today. All right, well, our Q&A sessions, I reckon we seem to have loads and loads of questions. So let's take a few for Richard first, and then we can maybe dial back to a couple left over for Scott.
Wanna kick off, Caroline? I think there's
Yeah,
a question from Ian Clark.
There are several questions from Ian Clark, so you clearly really captured his imagination here. Right, so the first one is about stage 1. So do you know why the reaction with the borane THF is slower?
No, is the short answer. Hang on, let me. There's a time course of the reaction, and actually T0 is at the end of the BF3 addition, and you can see that actually almost 50% of the conversion takes place during that BF3 addition. And I don't know if it's the BF3 acting as a Lewis acid to activate the amide, and then sodium borohydride attacking that. I don't know if it's forming some kind of mixed borane species with, you know, fluorines and hydrides and other things on the boron. No, I honestly don't know, but it's a really remarkable activity, and I do actually I often see reactions for amide reductions done using borane THF at reflux, and that really makes me nervous because the chances of making borane gas in the headspace, which is extremely flammable, is very high. And so I do sort of recommend this as a way of reducing amides if you want to take that forwards.
And his next two questions relate, I think, both to your stage 4 ternary phase diagram. So yep. First question was, what does the biggest triangle in the middle represent? Aha, yep.
So if you ignore the red line, the triangle at the bottom is the point where you have both the desired and the undesired out of solution. So it's an equilibrium mixture.
Okay. And does temperature affect crystallization in your ternary phase diagram? All questions already thought of.
No. It's a great question. The temperature, and so when we did this, what we I'm showing, I can't remember what, this is just a generic one. In the paper, it's got, I can't remember how many we put in. We did look at 2 or 3 different solvents at 2 or 3 different temperatures, and we did each of those was one slurry run, measure the eutectic point, and from there we could calculate the recovery from that at a theoretical 100% chiral purity. In reality, it was never that good, and we always ended up running it a little bit away from that and then doing a recrystallization, just because overall that's where we got the best yield. But as I said, this is a simplified system, it's an idealized system. There are some assumptions built into it, and so you can take it as a starting point, and it's a very, very good starting point. You'll always need to follow up with some experimental work to make sure it is operating as you expect it to operate.
Thanks.
Okay.
I'll hand over to Jason now.
Okay, yeah, so I've got a question again from Paul Colburn from Liverpool Chirochem. So he was mentioning that the dynamic kinetic resolution that you use to get to the chiral piperazine is very good work. We wondered whether
it's one of those pieces of chemistry I wish that I'd thought of it. It's absolutely beautiful piece of chemistry. Full kudos to the team in Italy who actually developed this.
Yeah, he was just wondering whether you tried asymmetric hydrogenation on, say, a parent pyrazine, assuming that was accessible, which I think is a pretty reasonable suggestion.
Absolutely. It was something we considered. I'm trying to remember. I think at one point we might have run a screen of chiral catalysts. I think that was quite a long time ago, a much earlier stage of the project, and at the time, just the technology was a little bit less developed, and so we didn't find anything that was really good. As a consequence, they developed this dynamic kinetic resolution, and because it worked so well, we just took it and ran with it, and we didn't go back and rescreen when we picked the project back up again.
Yeah, I can understand that. Yeah, I think the approach from Paul would probably work It's probably there's more developed
I've done on a
conditions now.
different project. I did look at asymmetric hydrogenation of pyrazines, and it wasn't trivial.
No.
But we did get some hits in that, completely different substrate. But yeah, we did get some hits in it, certainly.
Yeah, it's great. Caroline, do you want to continue?
There's a question here from Ollie Ring, who's one of our co-organisers. He's at AZ in Macclesfield, and he says, you touched upon incorporating agitation rate into your design of experiment. Considering particle size distribution, reactor, agitator, and impeller design can have massive impacts on reactivity of heterogeneous slurring chemistry. Can this data be considered robust following scale-up plant?
No, it was there as an indicator as to whether we might expect or how sensitive it was likely to be. So it was there as a quick look-see. So the stirring potentially has an impact on the chiral control in terms of the rate of the reaction. It also has a very major impact on the safety of the process. So as I mentioned, the sort of last or one of the primary stages in the chemistry is to add your BF3 dropwise to the slurry. And I did have one reaction where the stirring failed, and I managed to shut off the BF3 addition pretty quickly. But when I restarted the stirring, because there was sort of a layer of BF3, a BF3 rich layer at the top of the reactor, and then a sodium borohydride rich slurry at the bottom, when that stirring restarted, the two came together and there was quite a vigorous gas evolution. So from a safety perspective, it's really, really important. And it was actually written into all of our safety documentation. We actually had an interlock built in on our plant when we ran it in the plant, such that if the stirring stopped, the control software would automatically close the valve where the BF3 was being added. Coming back to the question, it's extremely valid question. We didn't believe we were in a place where it was sensitive. It was vital that it was well stirred, but actually beyond that, it didn't seem to be that sensitive in what we studied, but it was something we were planning to study more as we ran on relatively modest scale. It's 30 kilos, so sort of what's that, 500,000 litre reactors. As we scale up from there, we would absolutely want to study that in a lot more detail, and I've got many wonderful process engineers who were those who were working on the project at the time were, I think Frank was lining up to do that at the time when we stopped working on the project.
Jason?
Hi there. Yeah, we've got another question from Martin Sturmer from the University of Queensland. Welcome to you. A bit late in the day. Wondering if your borane reduction slowdown is related to some sort of diborane-borane disproportionation. Quite a good question.
Interesting. Yeah, I don't know. It must be said this was something I never got to the bottom of. Sooner or later, and it's something I'm not good at, I tend to get carried away and very excited about reactions and want to study them in enormous depth, and occasionally I have to rein myself back in, or one of my colleagues has to rein me back in to say, we've got something that works, just live with it. There's two things. So I think there's two things that are worth noting here. So this, so absolutely, majority of the conversion is during that that addition. There's also a little bump at the end here where you add the alcohol. And actually, if you do the quench much earlier, so you can do the quench at about 70% conversion, and it still jumps up to over 90% conversion. And so I think part of it might be solubility. When you add the alcohol, you're just increasing the solubility. You're helping because it's quite a slurry at that point. So it may just be that it's helping turn over some of the material into solution to allow it to react. It may be again that as the alcohol comes in, you are forming some other boron species which may be more active. I'm speculating here. I don't know, but there's definitely something going on there. I would be fascinating to look at it in more detail. I'm just desperately trying to find the time to do it. It's a great question, and thank you for staying awake this long. I'm really glad I haven't put you to sleep.
We've actually got quite a few questions here, which were for Scott. Scott, still there?
Excellent.
Yep, I'm here.
Right. So Kate Gilbert says, did you find success with the palladium catalyzed aryl sulfonyl fluoride synthesis on a on any heterocycles? Sorry, it cut off the end of the question.
I'm not sure what you mean.
Oh, so I think could you use any heterocycles as substrate? Do you mean, I think you might mean aromatic heterocycles.
Okay, can you see?
Yes,
Yeah.
can.
All right, so in the original sulfonyl fluorides, we were able to use a number of heterocycles in the sulfonyl fluoride synthesis. When you start getting into the vinyls, I mean, I think the substrates themselves, as you're starting with vinyl triflates, kind of precludes using heterocycles themselves. I mean, the closest we got was this indole, and there's a couple of other ones I believe I dropped out, but as far as sp3 hets, they were a number that we did use. I hope that answers the question.
Yeah, I think that's pretty good coverage. We've got a follow-on question. I'm going to have to see if I can pronounce this. Masakazu Atobi, I think he's been on a secondment at York University from Japan, from Asahi Kasei Pharma, and he's curious about your stability of the sulfonyl fluoride in storage, and whether you've done any stability test on long-term storage and moisture. I think that's a good question.
That is very good, very good. I think we've had quite a bit of history of the sulfonyl fluoride being stored for long terms. In fact, I have a flask of this that's been around for probably a couple of years now. That actually we used to get this crystal structure. It's still under the hood. You know, no nitrogen, no nothing, just a round bottom. As far as a broader sense, I think in general the sulfonyl fluorides are much more stable than sulfonyl chlorides. We haven't done any specific long-term studies themselves, but I would, from what we've seen and just the commercial sulfonyl fluorides that we can get, they're in general much better than sulfonyl chlorides, but I don't have any specific data to indicate that, you know, as far as humidity and temperature and such.
And one last question from Colin McKinnon, who says the favored catalyst looks slightly unusual. Is it commercially available? And do you need rigorous oxygen exclusion?
Amphos itself the ligand itself is quite, I think you can buy it. I tend not to, because you can buy the pre-catalyst as the palladium-2 complex from a number of sources, including Aldrich. And I personally like the Strem and the Johnson Matthey. They work really well. But the actual, the complex is very stable. I don't even bother with the nitrogen in filling the bottle with nitrogen or anything. But the naked ligand itself is quite sensitive to air. But yeah, this is commercial, and this is one of the reasons that we like this this kind of reaction for a lot of our SO2 chemistry, is because you're only adding just a few reagents to each reaction. There, you can do them out in the open. There's no glove box, you can even run these reactions without nitrogen and still get very comparable yields. They're perfect for library setups, and in fact, when we do a lot of SO2 chemistry, this sequence here is shown for the sulfonyl fluorides, but the intermediate is the same here, whether you want to get a sulfonyl chloride or a desulfonyl coupling or any of the other SO2 type chemistries. You can still access the same intermediate with this this first stage reaction here. So this one has kind of become our favorite go-to sulfonate generation, at least in discovery. And I have to ask around about process chemistry. That's a bit different, but yeah, we use this all the time, and it works really well. And you can set it up on the bench with no real concerns for any argon or anything like that.
Lovely chemistry, guy.
Very good. Yeah, it's really been a tour de force on all the chemistry today.
Thank you.
Yes, sir.
Thank you very much to Scott and Richard. And also thank you to my co-presenters Jason and Caroline for their hard work today. So yeah, I'd just like to close out by reminding you, if you were here earlier when I was talking about it, about next week's webinar, which is going to be a special one. It's going to be a showcase of some of the winning teams of the 2020 National Retrosynthesis competition, organised by a joint effort between the SCI and the RSC. So these three teams from Leeds University, from GSK again, and from Eutectic are going to be presenting their excellent routes towards this compound, Nidiserpinoid A. And lastly, just thanks again, everybody, for this week's webinar. See you again next week.